Good afternoon. Well, I'm sorry, good morning. I always make mistakes, but some people are leaving the room and you're going to miss the best part. We have already mentioned how we feed the soil. I'm going now to continue with the idea of explaining concepts. In the first symposium, we tried to tell you what we know, what we are certain of about how the soil works. Something we are not going to revisit in this symposium. But based on the information of previous talks, we have now certain indicators of soil quality that allow farmers to fine-tune their management practices. So based on the first talks, we are moving to a more hands-on ideas. We may say that the microorganisms that are in this drawing, these are the microorganisms described by Leonardo Erichmann. There's a lot, there is some diversity of microorganisms that face the challenge of being in that soil or going closer to richer areas in the soil, which is the rhizosphere. That's the area around the roots, where roots exudate a big part of the photosynthase that transform through photosynthesis. This rich area of the soil concentrates most of the microorganisms. And by studying these rhizospheric microorganisms, we can isolate many different microorganisms and study their relationship with the performance of the plant. And this is the topic of Mr. Valverde's presentation. This interaction of microorganisms with plants and microorganisms isolated in the lab that are cultured, we will go to the possibility of industrializing those microorganisms. This industrial plan seems like a Game of Thrones crown, but that's how I draw. I am now updated. Now I talk about Game of Thrones, not about Star Wars. I'm more modern now. So this use of microorganisms show the interaction of all the actors, all the players in this conference, farmers, scientists, and companies. This interaction is crucial. We have to promote it, all of us. We study soil biology, we study soil microbiology because we want not to study it in a laboratory. Our interest is not biology and microbiology. I am not interested in soil biology, in natural soil biology. I'm interested of soil biology in farming. And in order to understand it and to manage it, we have to study it. And in order to study it, we have to study it in real cases where farmers are really working. That's why we need this interaction. That's why we need APRESID and APRESID support to science. And the results of our tests are used by companies that transform and produce products that are used by farmers. So think about your questions. So we get them all together, and by the end of the symposium, we have a Q&A session. Thank you, Luis. And now we have Claudio Valverde, a head professor of the University of Quilmes, an independent researcher from CONICET, and he runs a laboratory of soil interactions and develops projects about interactions of soil and plants to promote vegetable health. Claudio Valverde is taking the floor. Thank you. Thank you, Aprecid. Thank you, Luis, for the invitation to be here today. Thank you to everyone present in this room. It's a pleasure to be here. I have to continue with this talk. I'm going to focus on a subset of microorganisms that have been mentioned by Luis and Leonardo. And these microorganisms that for about 40 years were called PGPR. Many of you probably know this acronym, PGPR. This is a Rizobacteria. These are growth-promoting Rizobacteria. This has been in the market for about 40 years, and we have more and more feedback from the laboratories to validate the concept. So first of all, let me detail the Rizobacteria part of the acronym, which is the subset of bacteria in the soil whose habitat, whose preferred niche to reproduce and live the root system of plants. This is the compartment that is so crucial for Rizobacteria. This happens below the surface, and many of the processes that occur here define the health of our crops. In order to focus on Rizobacteria, we have to zoom in, because these bacteria prefer this habitat, this special area of the root system, the rhizosphere. It is a layer that has only few millimeters thickness and goes from the epidermis. This is a crosscut section, and rhizosphere is around the roots. It is made of soil particles by exudates that come from the roots. It is a polysaccharide matrix that bring together these soil particles. The most important thing here is that we have much more life than the one we see, and we need to zoom in a bit more to see it. So we go into sub-millimeter scales and resort to certain technologies that allow us to see the microorganism in a natural environment. What we see here is a wheat root collected in the field, trying to be careful with the structure of the particles around the root. And we can see, using these techniques to check the DNA, we can check that near, in an intimate relationship with the roots, there are bacteria of filamentous nature.
These are actinomycetes and other bacteria that are depicted in blue here. These are the protobacteria that are tightly related to the root surface. These bacteria are what we know as rhizosphere bacteria. These bacteria are here because they found the way to take advantage vis-a-vis other bacteria that live in the soil. As Leonardo says, that huge asset of bacteria and microorganisms living in the soil. A part of these bacteria found the way to access the root and develop a number of activities I'm going to refer to now. This is not a unidirectional decision of the bacteria. Bacteria do not think. This is an evolution process. And this has been driven by the currency, which is the amount of energy and food, the amount of carbon and substrate feed that plants exudate through their roots. The amount of this will vary depending on the type of crop, the type of plant, but from 20 to 40% of the carbon that is assimilated in the air part of the plant is exudated through roots. This is a huge amount of carbon. Those of you who keep making a balance of this matter will also be surprised. And this happens because plants need to sustain, to keep the biological activity of the high number of microorganisms living in the rhizosphere. There are not only bacteria in the rhizosphere, because this is a very complex environment. Around the roots, we are going to find fungi as Oomycetes, Archaebacteria, protozoa, which are very important in the atrophic chain because they feed on bacteria. Arthropods, that's micro and mesofauna that has been presented by Camilio, and nematodes, which are also bacteria predators. So there is a significant biological diversity around roots in that window of a few millimeters around roots. Why? Because trophic chains establish, roots exudate a high amount of carbon, and that carbon is cycled by way of energy, and they feed other trophic chains. These biological groups are not there isolated from each other, but they establish natural interaction among them and with the root, with the plant. So in that, among those organisms living in the rhizosphere, we are going to have beneficial organisms for the plants. These are the organisms which the plant try to attract, and we are going to have neutral organisms and others that are undesired. These are the pathogens. From the interactions occurring between these groups and the plant, we are going to decide or determine the future of that plant when the seed is planted and the plant develops. That's why it is very important to analyze what is the biological situation in the rhizosphere and which organisms contribute to the growth of the plant and which don't. How much genetic material we have of each of them. We have to consider the abundance of each main groups. The numbers I've included here that I've highlighted as exponential numbers of base 10 represented the amount or the quantity of genetic information that we have next to the rhizosphere, next to the root in the rhizosphere. For instance, about 10 to the 10th genes of bacteria. So you will see that the group of bacteria are the ones that more information contribute to rhizosphere. So when one speaks about a plant, and specifically we are considering what happens underground, plant roots are not the only organisms, but rather they are surrounded by a second group of information data, and this is the information provided by the bacteria. This is the rhizosphere microbiome, using the term that Leonardo has just presented a while ago. So far I have been presenting to you who are or which are the participants, which are the microorganisms, specifically bacteria that live in the rhizosphere. They are colonizing the rhizosphere. But these microorganisms, specifically bacteria, are there because of the first part of this acronym, plant growth promotion, because the plant had found a way to attract microorganisms that promote its growth. So plants have learned to recruit from the soil microorganisms that will provide functions. Thank you for the previous concepts, because there are many things in common. We analyze these at different scales, but there are common concepts. Plants attract functions. What are these functions? Microorganisms that are able to improve in some way or other through a biochemical process the access to nutrients. The best known case is nitrogen through nitrogen fixating bacteria. But microorganisms help access to other micronutrients, phosphorus or iron, or even micronutrients. This is another function that plants try to find in the bacteria they recruit. Another group of microorganisms, of bacteria, are able to synthesize and to provide the plant with hormones or substances that are very similar to plant hormones, or they enable interaction with the hormonal signaling of the plant, and in this way they contribute to the development of the root systems, and it also allows keeping the metabolic activities and phase stresses as the water stress or salt stress. So this helps the root system to tolerate biotic stresses. And there are another group of microorganisms that are recruited because of the function related to the health of the plant. These microorganisms are able to conduct biocontrol of pathogens. They limit other microorganisms as fungi that create diseases and therefore cause the growth to be reduced or slowed. So plants and crops have, by evolution, developed the capacity to recruit from the soil sets of bacteria that would preferably colonize the rhizosphere, and they do this because they contribute with a number of benefits for these plants. That's why these PGPR organisms are called as plant probiotics, just because we make analogy with the concept that has been developed for human beings of probiotic microorganisms, which we have in our intestine, which help us take advantage or utilize the vitamins and other growth factors, and it also helps us limit pathogens. There is a very close similarity between human probiotic microorganisms and plant probiotic microorganisms. In this part of the symposium, the focus was on can we access to these microorganisms? Can we use this in farming processes, which we all know aims at being sustainable over the long term. So we want to keep productivity and food production in the long term. And we want to reduce the dumping or the input, the chemical input into the soil. So there is a very significant focus on these microorganisms, because if we can have access to them, then we will have the possibility to manage them and to have an impact on crops. And in fact, Gustavo will make a specific presentation on the industrial use of these microorganisms. And so a fraction of these microorganisms that live in the rhizosphere can be isolated in the lab through microbiological methods I'm not going to dwell into now, and they can be purified, they can be analyzed, they can be genetically characterized, they can be identified, they can be biochemically analyzed, we can find out what the functions are, and we can analyze their relationship with the plants under study and how they impact plant growth. And so we can conduct a screening process, selection, test under controlled conditions, and though we enable a field trial stage where we can use higher scales, larger scales, and so they can use them as bio inputs, as a tool. I won't go further into detail because Gustavo will give us more information. Which are these functions we can access to? These are shown by bacterial groups. You are going to recognize that they have their own history, because rhizobia are bacteria, microorganisms that have been used for a longer time in Argentina and in different parts of the world for different legume crops. The idea of this group is to help access to a renewable nitrogen source, as is the air. But recently, other bacterial groups have been introduced, Azospirillum, Pseudomonas, and also Bacilli, and other taxonomic groups that contribute other functions and other characteristics for the development of root systems. For instance, the promotion of root formation, tolerance to biotic stresses, and access to other nutrient sources as phosphorus. This question mark here is on purpose because the huge diversity of bacterial microorganisms in the rhizosphere is really impacting. It's not as wide as in the soil, far from the roots, because we are talking only about a set of bacteria, but really it is impressive. We still don't know how many there are, and there are a huge number of bacteria that can be cultured and has not been analyzed yet. So there is a great potential there. As part of these bacteria can be cultured, this gives us the possibility to develop strategies to generate inoculants or agro-inputs that will help us provide more than one function in a single product. Also, the fact that they can be cultured and analyzed in the laboratory gives us the possibility to use not only the microorganisms themselves, but also their metabolic products, for instance, hormones. And there is another way to walk along, which is will probably be developed in the world, which is the genetically modification in bacteria in order to bring together interest functions and microorganisms that can be easily handled, that had a good stability in the seed and in the soil. So these are things that can be done with culturable microorganisms. I will share with you what we have done in our laboratory lately. Because of our participation in the soil biology project that was co-funded by APRESID, we were able to meet our objective, which was to evaluate the impact of good or bad practices under no-tillage in different Pampas regions. Our objective was to evaluate the impact on a specific group of bacteria with probiotic capabilities, such as the Pseudomonas. So we had to count how many bacteria we found in the different root and soil samples. And so we were able to isolate them and have them available in the laboratory. So together with another group, in collaboration with another group, which was analyzing pathogens that had incidence in the same plots, we challenged a set of fungi pathogens with the Pseudomonas bacteria that we had. What was the purpose of this? In this petri dish, we distributed the fungi all through the dish, and so we replicated each of the bacteria that had been isolated. And then we allowed them to develop, to grow in the same laboratory dish. So we were able to identify bacteria that inhibited the pathogens. So this helped us identify a significant number of bacteria with this capability, and then we made individual challenges with these bacteria, with each fungus, and we were able to quantify this inhibition. So we were able to obtain a panel of 20 isolates, all Pseudomonas, isolates that were taxonomically characterized, very diverse. There was no relationship between them. This is a way to look at diversity, the pigmentation that they produce in the culture media. These microorganisms were isolated and they were analyzed with the objective to find bacteria that inhibit pathogen fungi. But we didn't stop there, because it is well known that a bacteria that lives in the rhizosphere and that has probiotics function does not have a single mechanism or a single beneficial characteristic. It can have more than one. So in the lab, we conducted an analysis of each of these isolates in order to find out what was the power or what was the quantifications they showed of each of these traits related to root system development, its potential for growth promoting, or a fungi antagonist, or the capacity to produce metabolites. And we also analyzed a number of factors that have to do with the colonizing capacity, root colonizing capacity. So we made a comparison of the 20 isolates, and we came up with this diagram that shows each of these rates or indexes for this bacteria. So we found that at large we can divide them in two groups. Ones are in this sector which have a higher biocontrol capacity, and these others that are represented in this part of the chart that has a potential for root development.
To lesser or larger extent, they were identified as potential biocontrollers of pathogens. That's how far we got in the lab. But we're still interested in see how these microorganisms behave in the field, because we are not lab rats. So we want to see whether these microorganisms can be used as bio inputs. So we produce a field test. We selected 10 microorganisms from the collection with biocontrolling potential, others with direct growth possibilities or potential. We inoculated this in weed seeds together with Rizobacter, and in 3 different environments that you can see the different weed yields. In these different environments, we have Pergamino that showed the highest yield. Junin on the right shows better quality parameters. What we saw is that despite differences in yields in each of the environments, that is of course dependent upon the environment itself, this number of bacteria, when they were inoculated in the weed seeds, promoted yield compared to control, I'm sorry. So we have 4 examples here. In 3 with biocontrolling potential and 1 with growth promoting potential. If you check the panel of results on the bottom, you see that the relative increases in wheat yield were more important quantitatively in the site that had the lowest potentialities in terms of yield. If you need more details, if you need the statistics, the methodology, all the data, tomorrow during the poster, Bettina will be showing the results. She produced the collection of bacteria, and you can go into details with her. Just to end with my presentation, because I'm running out of time, I would like to leave a comment. You have the microorganisms in the rhizosphere, some that cannot still be cultured in the lab in a simple way or a simple manner. These do not grow in the lab, and they are hundreds of thousands of species. But there is a good news. Luis mentioned this, and our colleagues too. These microorganisms can be directly manipulated, and that is the good piece of news. There have been many sound studies that show that good practices, what farmers do on the soils, are crucial to define the populations that will populate the rhizosphere. And that, of course, impacts on growth and yield. So the message is plants, mainly the roots, are not just plants, but they are microorganisms with very close relationships that contribute to growth and health. And the most interesting thing for us and for sustainable agriculture is that they are accessible. The functions they perform are accessible, and we can exploit them. We can use them to define planting practices. And then we need practices, and we have to study practices further, because everything in the rhizosphere can also be impacted on through good practices, and that's where farmers play a crucial role. So thank you very much for your attention, and I'm sorry for using more time than allowed. Thank you, Claudio Valverde, for your presentation, and now we invite Gustavo Gonzalez, Anta. Before I introduce him, the questions that you're making to the phone number that is on the screen through text messages, please identify who you are addressing this question to so that we can handle the Q&A session better. Now, we introduce the next lecturer. He was graduated at the University of Buenos Aires, agricultural microbiology. He's a researcher at Rizobacter, Argentina. He teaches microbiology in the University of the Northwest part of the province of Buenos Aires. Gonzalo Anta. This is Gustavo Anta. Well, thank you very much for the invitation. I have to try to explain how we can transform this good science into applied technology. That's why the name of my presentation focuses on checking the impact and evolution of microbiology technologies on crop productivity and health, not only from the company perspective, but from a shared perspective between the academia and companies to get the best results in the field. When I started preparing my presentation, I wondered which could be the most important things to mention. So maybe if I could summarize the answers to these three questions, I would be more than satisfied. The first question is, what microbiotechnological transformation have occurred in the 21st century? For those of us who were born in the past century, we have already been in the 21st century for 16 years, and the evolution in terms of knowledge, both in the industrial part of the chain and in the academia, has been phenomenal. And it has accelerated a lot in the past 5 years. So I'm going to discuss these transformations. The second question is, okay, we've had transformations, but which have been the impacts on production based on these new microbiotechnologies? How have these helped us be more efficient? And the third question is share with you the challenges and suggestions for the future. We are working in a close relationship with academia to get these possibilities. It's important to highlight that in the past century, 1990, 1995, it was difficult to link the interest of the industry with the interests of the academia. But I believe that the interaction, this strategic association or partnership, academia-industry, has progressed a lot. We have agreed on protocols to approach different issues, to take to the lab our concerns and get from the lab answers to bring them to the fields. Then we defined the establishment of microbial functions in order to see how to increase crop nutrition with phosphorus, with N, with K, or how to control different diseases, different pathogens by using microbiological tools. And number three, which is also very important, is the synergy between basic knowledge, basic scientific knowledge, and understand the why and the main reasons why certain things happen in the plots. So this is the interaction between the academia and the industry that has proven to be very, very important. We can also say from the industrial point of view that we need to improve, and how we can improve technologies to make field management practices easier. So in the interaction with universities and research centers, we have worked on microorganisms survival. Late last century, it was hard to find papers focusing on how to keep microorganisms alive in a recipient, in a container. But currently we can do that. Thousands of microorganisms in a microliter. 10,000 million microorganisms that can be in 1 millimeter of an inoculant. It's 2 times 10 times 10, that is 20,000 million microorganisms. We have the technology to ensure 3 times 10 times 10 per milliliter, which is equivalent to 1 gram of soil. So in this rhizosphere, in this soil where we have so many microorganisms, having the capacity to incorporate many of the microorganisms that we want to have an impact is crucial, because we will see the impact in the crop, in the yield. So partnerships have been fruitful, because the question is not to have just many microorganisms, but to understand the impact on the yield, on the biomass production, understand the number of nodules and dry nodular mass to fixate N. And as you know, soybean requires a lot of nitrogen. We also wanted to focus on practical matters. This has to be achieved by planters and by the usual machinery we have, because if not, adopting the technology would not be so successful. We have completed many tests, and we continue to do so, because we have ended this paradigm of throwing a lot on a number of seeds. Currently, the idea is to have the microorganism per unit of crop to potentiate each seed in the row to get the highest possible yield. This has to be done under certain practices that have to consider different soils, different temperatures, different planting techniques. So our contribution, our practical contribution, is to check the interaction between the seed, the machinery, and the product we offer. And today we market products that have already been tested in all these areas that I have just mentioned. So which have been the technological leaps produced in the 21st century? I've been checking certain turba inoculant data. Some companies have used only one kind of inoculant, and that led to technical complexity. It was difficult to keep it sterile, but today we have liquid supports, and thanks to what we know, or thanks to what we have learned from the interaction, we know that these peat-based inoculants we can reproduce in the reactor stress conditions. We can stress bacteria so that they send the signals that we need to place them in the rhizosphere to produce better crops. We have made a huge progress. We know that plants produce different things. We know it produces flavonoids, fatty acids, polysaccharides, lipopolysaccharides, and other molecules. And those molecular structures are not the same considering the abiotic stresses they are subject to. So today in the industry, we can introduce different elements in our manufacturing processes to produce bacteria and prepare them for real conditions in the field, even though we don't have the ideal temperature or ambient humidity. We can produce something that can survive under difficult conditions. In legumes, we have the PGPR. We have many different species. We have seen different functions. We have focused on some abiotic stresses, nitrogen fixation, phosphorus utilization, which are crucial to have good yields. And we have also seen that this is translated into biomass, into food for the different links of the atrophic chain. And this leads to a more sustainable system. And we have root developments that are impressing, both in wheat and corn, and relevant biomass production numbers. So the system works better, it is more sustainable in the long term, and interactions do not stop existing, but we can help them be better. Let me also tell you that since we have made much progress in biocontrol matters and in biofertilization, also in biocontrol, especially in biofungicides, there was a technological problem how to put these microorganisms on the seeds to help to have them control certain pathogens. Though there were many papers published on trichoderma, etc., it was difficult to achieve a formulation to be used in the production of seeds. After 7 years, we could find the proper formulation with the proper strain. We work with a certain trichoderma and other organisms, and when you go to the field, you see the impact on yield because pathogens are under control. This is another aspect where basic science and applied science complement each other and make the sector progress. This is not only about pathogens, it also applies to pest control, to weed control. This is one step before the other products because we still have to discover certain things. We have to understand certain behaviors under ultraviolet light, and we have to work on that more strongly.
But I said that we would try to answer to 3 questions. This was the first. What innovations were introduced in the 21st century? But what has been the real impact, or what productive impact did the new microbiotechnologies cost? These are some of the examples. Here, this is a bioinduction technology. The check is conventional inoculant. This have been conducted in different regions of the country, different management, different planters, different seeds, different climate conditions, and see how the association, academy, and industry was able to achieve a considerable projective impact to improve what was already there. We were not using an absolute check, but rather a conventional inoculant. Many times, the findings in the lab or in the growth chamber are great, but when we take them to the acid test in the field, results are not so clear, but we have been successful here, and we can say that bioinduction technologies working on recognition of biomolecular characteristics of plants and bacteria and interaction give very positive results. The same happened with the Pseudomonas fluorescens. We clearly see that the impact, productive impact in more in poor systems, in deficit systems, where there is absence of phosphorus or blockage of phosphorus, the impact on yield is very interesting. And in environments where we have less nutrient availability in the northern part of the country, Bolivia and Paraguay, or South Africa, where there are restrictions in nutrients, this type of technology helps a lot, not only because it increases nutrient availability, but it also improves root development versus phylohormones production. And when we go to the field, these are productive plots. I insist this is with a technology of the product. This is not a trial plot, but this is a quarter of a hectare in each plot. And when we use a biological tool that has been selected, identified, and characterized in the lab, but at the same time, it includes a significant amount of technology because it can be taken into practice, adding them to the conventional seed treatment. We have also obtained good results, and we see that the farmers are willing, because each of these technologies, induction, growth promotion, and biocontrol, have a much lower environmental impact compared to the use of conventional chemical products. Finally, although we are facing some problems regarding the technology to be used for the manufacturing of our end products, in the case of bioinsecticides, we made progress with isolating several microorganisms. Some are bacilli, other are Thuricienses, other are different from that. We use them with two pests which are really a headache. The case of Spodoptera, we see 100% control. In Rachyplusia, we see that control is close to 90%. So although there are still some technological difficulties, there is another way the industry is walking along in order to provide alternative pest controls. But we had three questions, and this is the last one, but the most important one. What challenges and proposals from the microbiotechnological point of view we have for the future? In this Congress, we are going to speak about climate change, as we did in the past. This is something that has come to stay. This will with us for good. Variations in temperature, humidity, strange winters because they are too mild or they are too strong, the same with summers. the same as plants have to adapt to new situations, microorganisms that we use for those plants also have to be adapted to the situation. Therefore, together with the Biopass project and the Leloir Institute, we are analyzing a number of microorganisms that are linked through the Pseudomonas that have an interest in behavior in stress condition. Therefore, in the future, when I speak about future, I'm talking about the next 3 years. I'm not talking in the longer term of 10 or 20 years on. We are exploring and working together with research centers in identifying and field testing of different microbial groups that can contribute solutions for the different problems. This is one of these cases. The second story is the fact that microorganisms work cooperatively. A single cell cannot produce a beneficial effect or a disease. There is a group of cells working, so it is important to be able to provide different solutions at the same time or different functions so that the seed is inoculated with different microorganisms. They will stay in the soil where they will have competition in space, in water, in nutrients. So the use of consortia and the impact that this has in production level is very important. This work has been conducted with different strains of soybean of different species. We see that some of the combinations achieve performances that are higher than others. This implies huge industrial complexity. However, results are very interesting. And so this encourages us to continue working in order to obtain groups of inoculants that contain a significant number of strains and different species, because we see that this is effective and this will help us face the challenges we have ahead of us. Through Biopass or Biopass and through the association with other research groups, Gonzalez, INDEAR, UNOVA, companies such as Rizobacter and Bioinsumos, we are proud of drafting a paper together with a very important magazine, Nature. We are convinced that knowing why there is an interaction between microorganisms and plants, we conducted this work in 11 different locations of soybean crop. We analyzed which were the microbial composition of each of these plants. And the most interesting thing is that we started to isolate those microorganisms in order to know whether we are able to have a more favorable impact on the production system. As Leonardo was just saying, this means integrated the information of plant microbiologists. We are working on that. We are committed to that, and therefore we are interacting with different research groups to make progress along these lines. I loved the fact that Claudio referred to the use of genetically modified microorganisms. I'm running out of time. I know that. I will close here. 12 days ago, something that should really honor all of us. There are many people here used to work with genetically modified plants and animals. We have obtained the approval of Conavia after a long work of submitting information to conduct a controlled trial with a microorganism that does not fixate nitrogen but has interesting agronomical traits. This is a Pseudomonas. that has been added this function of fixating nitrogen. And this opens a whole new window specifically for grass crops. Imagine if we were able to reduce the amount of nitrogen that we use in grass crops to maximize yield, or if we were able to have a more effective use of those nitrogen when we plant wheat in Anta Castelar, we are only starting with the implantation of this crop. We are going to follow it closely, and we will analyze how long this microorganism persists in the soil and how it interacts. So this is a very important project that we have in hand. I want to give you this take-home message. In an altered world, I think that this association between industry and the academy is really productive. As Mother Teresa said, I do what you can't do, you do what I can't do, so together we can do very big things. Thank you very much.
A Gustavo González Anta por su presentación. Estamos ya sin tiempo.
I will ask Luis, please, to close this session. I just wanted to close first thanking the four speakers, and I would ask you to, after all this information, you incorporate soil biology in your heads of agriculture, in your head of farmers or advisors or industry people. We come from a very physical, chemical farming management, and I was considered the paradox here of an agrochemical company. We spoke about bugs, about pests, and they spoke about fungicides and insecticides. A short time ago, we were working in the farm. There was an advertisement of pesticides that said it doesn't leave anything on the soil, and we wonder what are we working for? And it's just the other way around. So I ask you to make an integration between physical, chemical, and biological management of soils. It is true that there's still a way to go and develop knowledge to manage soil biology in a faster way, as fast as it happens with chemicals and with fertilizers. But let's not forget that soil works or functions thanks to its biology. The soil is not a flower pot, it's an organization or an organism with biology of its own. Probably we can ask the questions you have over the break.